Now moving on to the biochemical reaction and identification of bacteria. So I told you the details of culture media which are used in the diagnostic laboratory. For example, depending on the hemolysis, you can identify it's alpha hemolytic streptococci, beta hemolytic streptococci, and gamma hemolytic streptococci. If it is an enteric organism, you have to McConkey's medium. If it is a lactose fermenter, it is Klebsiella, E. coli, non-lactose fermenters, Salmonella, Shigella fructis, and Pseudomonas. To differentiate Staphylococci from Staphylococci, we have to do oxidase test, oxidase and catalase test. Catalase test is very important more than oxidase test. Oxidase test is positive in gonocon. Now moving on to the biochemical tests which are very important. I will tell you the basics how to identify the organism in the laboratory. First of all, very important is clinical specimen, whether it's urine, pus, sputum, stool, CSF, various body fluids. So when it comes to the laboratory, urine it should be more than, if the processing is more than 4 hours, it should be refrigerated, and blood culture and everything should be incubated in incubator. So first and foremost, gold standard in microbiology is gram staining. So once you do a gram staining, you come to know organism which is responsible for the infection. It's a gram positive or gram negative. The gram positive and gram negative, essentially all the organisms grow on the blood agar. It's a gram negative, use McConkey's medium. So once you grow the organism in McConkey's medium, you can depending on the colony characters, you can identify whether it's lactose fermenter or lactose fermenter. I already told you lactose fermenters are equivalent Klebsiella, non-lactose fermenters are Salmonella, Shigella fructis, Pseudomonas, and Vibrio cholerae. Now, first I start with catalase test. A lot of students get confused with the biochemical reactions. It is, everybody thinks it's difficult to memorize it. I'll make it very easy. First is catalase test. What is catalase reagent? Hydrogen peroxide. And you take the loop full of hydrogen peroxide, put it into the broth culture and pour it on the colony. There will be bubbling. Hydrogen peroxide is reduced to water and effervescent oxygen. Now next is oxidase test. Oxidase reagent tetramethyl paraffin diamino dihydrochloride. So very important to differentiate between Pseudomonas and Enterobacteria. Pseudomonas is oxidase positive, all Enterobacteria are oxidase negative. You prepare 1% of freshly prepared tetramethyl paraffin diamino dihydrochloride, rub the colony on the filter paper, change the color, or pour over the colony, change the color. Please remember, oxidase positive is Pseudomonas and Enterobacteria are linked. Now there is an IMEC test, indole, methyl red, Oaks-Prosker, and citrate. Indole test is conversion of tryptophan into gas indole. Grow the organism overnight in peptone water. Pour coax or early reagents, and the formation of pink ring suggests indole positive. E. coli is indole positive and Klebsiella is indole negative. Oaks-Prosker, methyl red and Oaks-Prosker are detection of complete formation of acid or intermediate product called acetyl methyl carbonate. Grow the organism, glucose phosphate broth, add MR reagent, change in the color suggests MR positive. The same broth, use VP reagent, change in the color VP positive. If organism is VP positive, it's MR negative. If the organism MR positive, VP negative. Citrate, ability of organism utilize citrate as the only source of carbon. are sole source of carbon. The Kosher citrate is liquid medium, cement citrate is solid medium. Change in the color or growth suggests citrate positive. Imic, in short, it is called as E. coli is plus plus minus minus and Klebsiella is minus minus plus plus. So there is another media, composite media, which is very useful in the laboratory. It's used for identification of bacteria. Triple sugar iron agar. It contains 3 sugar, lactose 10 parts, sucrose 10 parts, glucose 1 part. And it also detects hydrogen sulfide production and fermentation of 3 sugars. So depending on alkaline present, acid but and alkaline sand, glucose is not fermented. Acid but acid sand, both all 3 sugars are fermented. Bubbling shows gas formation. Blackening shows estrase formation. One more medium which is commonly used is urease medium. The ingredient of urease medium is, remember, PAPU, peptone, agar, phenol red and urea. The organisms splits urea into ammonia and carbon dioxide. There is a change in the color of the medium. The organisms which are urease positive are highly processed within half an hour. Positive is protease. Please remember these reactions: catalase, hydrogen peroxide, oxidase, tetramethyl paraffin diaminohydrochloride, the indole, conversion of tryptophan into indole, coax and enriched reagent, methyl red dose grow in glucose phosphate broth, pour MR and BP positive. If the organism BP positive, MR negative, MR positive, BP negative. Citrate, ability of organism to utilize only source of carbon, or citrate as the only source of carbon. Then triple sugar iron agar, 3 sugars, sucrose, lactose, glucose, 10 parts, glucose 1 part, and ferrous ammonium sulfate, and change in the color, acid but acid medium, all the 3 sugars are alkaline medium, alkaline slant, only glucose is not fermented. Blackening shows estrase fermentation. Urease, change in the color, it splits urea. Organism utilizes urease enzyme is produced by mostly by protease organism. It splits urea. Urease splits the urea into ammonia and carbon dioxide. There will be change in the color. Highly positive, urease positive organism is protease. You remember punch. Punch, Tata car has come, punch. The protease, urea plasma urelyticum, nocardia, PUN, urea plasma urelyticum, nocardia, helicobacter. All these are urease positive organisms.